Okay, our next section of evolution, we're going to talk about the evolution of populations, which is microevolution. Now, what we're looking at here is a population of penguins. So a population is a group of organisms of the same species that live in the same place at the same time, interact, and interbreed. So genetic variation is essential to the survival of a population or the survival of a species. And when we're talking about the genetic variation, we have to go back to some of our terms from genetics. So genetic variation leads to phenotypic variation. So when we're talking about all of the genes or all of the traits of a population, we're talking about that population's gene pool. So when we're looking at this gene pool of frogs, we see that they have coloration, they have webbing in their toes. All of those things would be characteristics of their gene pool. When we look at the marsh grasses, we do see coloration, but we don't see webbing of the toes. They don't even have toes at all. So they have different genes, different characteristics in their gene pool. Now, what this is, and I didn't want to spend a lot of time on it, but I wanted you to have a little bit of an idea about it, and I've got a link here that I'll put for you all to look at, is something called the Hardy-Weinberg equilibrium. And there are certain conditions that have to be met in order for this to occur, but I want to just kind of throw this out there so that you can look at it, have a little bit of knowledge of it, at least have seen it. But we look at these organisms here, this little group of frogs, this little population of frogs, we could see that they can be green or they can be brown. And we could see that there's a big G for green, so we know what that means. It's dominant, and a little G for brown. And we see a little bit here about the frequency of these alleles. So that might be something that you would hear in evolution is frequency of an allele. The frequency of the allele for green, the frequency of the allele for brown. And when we look at this population, this particular population of frogs, we could definitely see that more of them are green. I just took this from your online textbook, but it goes through the same thing with the fish, using their tails as what we're looking at. So look at their tails, big T, of course dominant, and little t for recessive. So having this tail that looks kind of scooped in versus one that is more solid. And then here is the Hardy-Weinberg equation. So the P is the frequency of the alleles for the dominant allele, or homozygous dominant. The Q represents the frequency of the recessive, so homozygous recessive. And then the 2PQ, that is the dominant and the recessive, so that's the heterozygous individuals. I also put a little bit of information on here that you can look at through Khan Academy. So there's a link that you can go to and do a little bit of practice with Hardy-Weinberg. But again, we're not going to spend a lot of time. I just wanted you to see it, see what it looks like, to know just a little bit about it in case you hear about it again in the future. Okay, so where do these genetic variations come from? Now, most of these genetic variations are going to come through the gametes from the parents, so they're going to be inherited. And way back when we went through meiosis, we did a little lab on crossing over. So remember, we did this with the candy. So this exchange of genetic information creates chromosomes that are different from the originals from the parents. So this is what goes into the gametes that increases genetic variation. I've got a little link here in case you've kind of forgotten what crossing over is. Put a link, you can watch that video. And another source of genetic variation can come from mutations. Now, we didn't get to talk about mutations, but mutations are mistakes that occur in a gene sequence. And there are many different types of mutations. But if we're talking about a mutation that occurs in a somatic cell, a body cell, like in a skin cell, that will solely be my mutation. my problem. But if it's a mutation that occurs in a gamete, then that will be passed on to the next generation. So again, I've got a little link here to a video about mutations. So please watch this video from your friends, my friends, the Amoeba Sisters. So what we're going to look at natural selection in populations. But first, before we look at what natural selection can look like, we've got to look to see what's normal, or what normal looks like. So if we're looking at a population of whatever it might be, this is what you get, is this bell curve. So the highest frequency, the highest occurrence of whatever trait we're looking at is going to be right here in the middle, what's normal, what's average, so the mean, and then you'll have fewer at the extremes. Okay, so there are 3 different paths. So I've got a video here about Charles Darwin and selection. So please watch this video on your own. It's one of my personal faves. But here are the 3 different pathways. One is called directional selection, stabilizing selection, and disruptive selection. Now remember, all of these are natural selection, so human intervention, not a part of this at all. This is all about environmental pressures. So directional selection, just really quickly, is in favor of one extreme form of the trait over another. Stabilizing selection is in favor of what's average or what's normal. And disruptive selection is in favor of the extremes, both extremes. So we'll start first with the directional selection. And again, I have a link there. Please go to it. I would watch it right now, but Screencast-O-Matic doesn't allow me to record sound from the computer, only from my voice, unless I upgrade. Not doing it. Okay, so directional selection is where you have one extreme favored over another. And I've got a gazoodle of examples here. But this one has giraffes. So giraffes, we can see the blue line is after selection. This shows us that giraffes have these longer necks because it is a favorable adaptation. So the extremes, the short is favorable because you just kind of think about the background. They're able to, these long necks, they're able to reach more vegetation. Then they're going to have more food, they're going to be healthier, they're going to live, and they're going to pass on their long necks to their kids. That's just what it is. Now this is a slow process to start to see populations shift. This one looking at a group of mice in some type of woodland situation, and we could see all the different phenotypes, all the different types of fur color that there can be. And then we see, based upon this population, that as they've migrated into these woods, that we're now seeing these darker colors being favored over the lighter colors. So again, thinking about the environmental pressures, they go into this forest. These guys must be able to blend in more with their surroundings and survive predation. These moths here, and you'll hear a little bit about this in the video that I've asked you to watch, after the Industrial Revolution, that having this darker color was favored over this kind of peppery color, because with industry comes junk in the air, pollution, and then these guys were the ones that were more camouflaged and were less likely to be eaten. This example of drug resistance in bacteria. So it is favorable for the bacteria to have higher drug resistance. So antibiotics themselves have not been in play in the population since about the 50s. And at that point, there were no bacteria that had antibiotic resistance. But as time has gone along and different practices have not been followed correctly, then we tend to seeing more bacteria that are drug resistant. Today we call those superbugs. But some of that is attributed to people not taking antibiotics in the correct course that they're supposed to take them, or overprescribing of antibiotics. So directional selection, favoring one extreme or the other. Okay, the second one is stabilizing selection. And in stabilizing selection, we see that what is normal, what is average, is what is favored. So if we look at this one, we see these medium-sized lizards are favored in this particular population from the larger lizards or the smaller lizards. And again, it has to do with the predators. In this case, in the woods, wherever these little mice have roamed, then this color is favorable. So not too dark, not too light. So these guys blend in just right. And then this is the example from your textbook, plant galls, so G-A-L-L-S. And I stuck a little picture in there, and I've put a link so you can go and investigate a plant gall. But basically, it's a protective measure of a plant based upon an infection. The particular type of infection that I posted here for you is from insects, from bugs, but it could be fungus. A lot of different things. But basically the plant produces this kind of swollen mass of tissue to try to contain whatever the infestation is from maybe a parasite. But what we have here is the galls that are favorable to wasp and are favorable to woodpeckers. So wasp like the small ones, because when they start pecking in there and get into this, there's gall, there's probably some little fly larva, there's something tasty in there that they're going to eat. Same thing with the woodpecker. They prefer the large ones, so they're going to dig in there and they're going to eat whatever's inside. So in this case, it's favorable to kind of be right in the middle, to be medium sized. Now, yesterday morning, my puppy dog was going crazy at the front window, and I looked out there, and there was, now this one's not as pretty as mine, but there was a beautiful woodpecker with this big red mohawk pecking away at an old tree stump. And I didn't have anything near me to go take a picture of it, but I found a picture of it, and I found the bird call. Now, I haven't heard the call, but I did see that I think this is a pileated woodpecker was just plowing away at this old tree stump, trying to eat whatever's in it. No plant galls, just old tree stump. But very interesting, this guy. And then the last is disruptive selection. So if you look at this bell curve, it has definitely been disrupted. It's completely messed up. There's no curve left. So both of the extremes are favored. So back to the little mice, maybe it's favorable to be either really light or really dark. This situation could lead to these organisms being so different, could lead to speciation. So again, I've got a little video clip from your friends, my friends, Amoeba Sisters, for you to look at for speciation, or the creation of new species. And then the example from your book, I guess I'm kind of on a bird thing right now, are these lazuli buntings. I'm probably mispronouncing that, but hey, I tried. And these lazuli bunting males, they have to compete for females. That's what they have to do. And what's favorable is either to have these lightish brown or these bright blue feathers, because what ends up happening is that these males here that might be a darker brown or have some blue on them, these guys, these dominant guys, are going to really compete with them to get that female mate. So it's favorable. They're going to find a mate. They've got pretty feathers, and they're going to dominate, and they're going to scare the other ones away. And then these guys, little brown guys over here, just kind of slide under the radar, and when they're fighting off the dark brown or the brown with the blue, they swoop in and find themselves a mate as well.